Hello, welcome everybody. Welcome to this live stream. Today, specifically, we will be taking some time to talk about randomized control trial. Oh, some weirds. So let's get started. And what I'll do now is to put a link. Some of you may see the link. I'm going to put a link here in the comment on the specific paper. And typically, I like to choose JAMA Network Open because first, it has a pretty good quality studies, and second, it's free access, open access. I want people on YouTube have access to the articles we are looking at. All right, I'm going to turn off myself, and so that there's more space for this. All right, let's get started. So this is a randomized control trial. Why I pick one specifically a randomized control trial is because we often do journal clubs choosing these sort of articles. And it is important to understand the different sections, the methods, and the results so that you can do a proper journal club and also critically appraise the paper. So what I have here, one is the original paper. I have the supplement. And I also have how I like to look at research paper. And I'm going to go back and forth so that you can see how I walk through this whole paper as a reviewer. And when I'm preparing journal club, this is what my process as well. Okay, so let's get started with the title because title gives us a lot of information. Number one, title, an enhanced strategy for daily disinfection in acute care hospital rooms, a randomized control trial. So now we know it is a trial, and then we can already know they are talking about specific setting, acute care hospital, and the intervention is daily disinfection. So I like to get started with the abstract because it's the most concise way. It gives you the key information. And if you find that this is an important article or interesting article, then you can move forward. So what is important? Why is it important? Environmental contamination is a source of transmission between patients, healthcare practitioners, and other stakeholders in the acute care setting. And the main objective, the specific aim, is to compare the efficacy of an enhanced daily disinfection strategy versus standard disinfection in acute care hospital rooms. Now, the next section is where the money is, the design, the setting, and participants. So this RCT was conducted in acute care hospitals at Duke Hospital from November 2021 to 2022, a short period of time, 4 months. And these rooms were occupied by patients with contact precautions. So they chose a very specific population. So it's not just any rooms that we can go in and out, it's those with contact precaution. It means somebody goes in, you have to wear a bunch of gowns, you have to wear gloves before you can get in. And these room surfaces were divided to two sides, right versus left, allowing each room to serve as its own control. And each side was randomized 1-to-1 to the intervention group or control group. So this is a very interesting design. So they take one room and split it to two sides. So right side, maybe they use the cleaning product, and the left side is the standard, and allowing it to become the control. So if you can think about it, if you have two separate rooms, maybe one patient in one room, another patient is a different room, then you may have the risk of the control not being the same. Maybe one patient has a different bacterial flora, another one has different. So this is quite an interesting way to think about, when you design trials, how do you make sure everything else is equal? So here, only one room, and they split half side. So what is the intervention? The intervention was this solution, some special wipe, ammonium salt-based 24-hour continuously active germicidal wipe, and is applied in addition to routine disinfection. So they did clean regularly, but added this extra 24-hour wipe. And the control group received no intervention beyond routine disinfection. Okay, so that's reasonable. And now the next one. We know the intervention. What is the main outcome? Because if we don't know the outcome, we don't know what outcome, we don't know what's end goal, right? So that's the most important. The primary outcome was the total contamination as measured by colony forming units on the bed rails, over a bed table, and sink on study day 1. So I can assume because they are looking at a 24-hour wipe, so they can look at the outcome after 24 hours, which is study day 1. And specifically, they're looking at some sort of the contamination. And then secondary outcomes, proportion of sample areas with positive tests for clinically important pathogens. And so these are a bunch of bacteria that are dangerous. MRSA, VRE. I'm going to use abbreviations here because these are very common. Yeah, carbapenem-resistant enterobacteria. Even I have trouble pronouncing this word. So basically, they will also want to look at other bad pathogens. To make sure that there is no, so the next thing is the similarity in baseline contamination between sample size on study day 0 before the application and then after day 1. So sometimes what can happen is, what if you, before you even do the intervention, there's some contamination already. So they want to look at the baseline before you do the intervention, what happens. So they are doing the primary outcome, which is the main contamination, but they want to make sure underlying before you even start, there is they want to make sure there's similarity in baseline contamination. Okay, now the results. So they had total 50 study rooms and 50 unique patients. So 50 patients, and the age 61, 26 men, it looks like half, with contact precautions enrolled. 82% were actively receiving antibiotics. 78% were bedridden. Interesting. So that means a lot of them are not really moving around. They're probably sitting in bed for a long time. And 56% had active infections with study-defined clinically important pathogens. And they found that on study day 1, the median CFUs for intervention group, so the one that got that special wipe, was lower than the control group. Okay, so 3,500 CFU versus 5,200 CFU. And on day 1, the intervention site was less frequently contaminated with a clinically important pathogen compared to control site. So this is quite promising, but let's look at the conclusion and then we dive deeper. The results of this RCT demonstrated that a quaternary ammonium salt-based 24-hour wipe could decrease the environmental bioburden in acute care hospital rooms compared with routine disinfection. The findings warrant a large-scale RCT to determine whether enhanced daily disinfection strategies can decrease patient acquisition and adverse patient outcomes. So I want to bring your attention to these last 2 sentences. So when we think about results, right, and we want to make conclusion and want to apply it to our patients, we sometimes, it's a positive finding. Positive finding, yes, they reduce the bacteria, but does it really change outcome? And so here they also already brought up some limitation. Okay, yeah, we see that it reduces the contamination, it reduces the number of bacteria, but we don't know at this point whether it can decrease the patient acquisition and adverse patient outcomes. So next, let's dive deeper. So now I'm going to jump to this sheet to show you briefly how it's very intuitive for me, so I don't go through this all the time. But when I'm reviewing paper, I actually look at the title, make sure, is it clear? Is it are they trying to, does it contain any unnecessary description? And abstract as well. Does it include enough information to stand alone? Then now, introduction, I want to make sure it justifies this research. Okay, so let's go into the introduction and see, do they justify doing this research? I'm going to skip right here because I've read through it. In general, they say this liquid chemical disinfectants used in healthcare settings are highly effective. So at baseline, it's really effective. And disinfection occurs immediately after disinfection application, but The problem is surfaces quickly become recontaminated. So yes, it works, but the problem is it gets recontaminated. And so one potential solution to decrease the risk of contamination is through continuous disinfection using technologies that have constant and long-lasting disinfectant action as opposed to only when applied. So now, as you can see, okay, they are using an intervention where the wipe is 24 hours. So it's a bit more long lasting. And now we can go to the aims, the goal of the study. We perform an RCT to determine the efficacy of this enhanced daily disinfection strategy compared with standard disinfection in acute care hospital. And they want to see whether if there's efficacy immediately after application and for 24 subsequent hours. And their hypothesis was that this use of enhanced disinfection compared to routine disinfection would decrease environmental bioburden in hospital rooms. So that is what they start off with. Okay, now let's go to methods. Okay, can we trust it? Can we trust the results? Before we trust the results, let's make sure the methods are accurate. Okay, so I'm going to jump back here. When I look at methods, I'll think first, study design. Is this the right study design to answer the question? Now, they want to basically compare two different strategies. So yes, absolutely. Randomized control trial is the right type of study design to answer the question. Now I move on. Population. What's the inclusion, exclusion criteria, and any potential bias? Okay, so let's read this a little bit more. Here was a summary. They said we performed this in a Duke University hospital, 1,000 patients, a short period of time, and all patients. Right. Let's look at the inclusion exclusion criteria. All patients were occupied by adult patients, 18 and older. Study rooms were identified by routine review of bed flow data, which is good because you don't want a special type. You want a regular clinical setting where depending on how the bed needs to be filled, they will fill it. Rooms occupied by patients with contact precautions were targeted for inclusion. Rooms were excluded if the patient was discharged before samples from study day 0 and day 1 were obtained. Okay, that's a reasonable inclusion exclusion criteria. And now I think this one we mentioned it before. 3 areas in the study room were split in half for the purpose of study: bed rails, the table, and the sink, left versus right. And then they sample areas size in each room were randomized 1-to-1, half side. And so overall, they evaluated 3 sample areas and 1 each side in each study group. Okay, so here, now let's talk about blinding. All the clinical, lab, and statistical staff were blinded to the randomization triple blind study. However, the study team members, including A and B, who applied the disinfectant and collected environmental cultures, were not blinded to the study group. Okay, so this is a very important area. Why do we need to blind? So imagine you have the people, the clinical people know that you are doing the study. They know then they may not want to touch the railing. Maybe they change their behavior. Maybe they clean a bit more. And then lab, you never know, right? So you want to blind as many as possible. So now they did not blind the study team. Is it reasonable? Sometimes it's difficult to blind everything. But if you think about it, they are merely getting a swab from each site. So at the end, they still need to send to the lab for culture. So fairly, I would say they are doing a pretty good job, blinding all three different groups. Right now, study protocol. When it comes to study protocol, this is really important. The most important thing about a randomized control trial is not really the analysis. It's how you conduct the study. Because the cleaner it is, the more organized it is, the more replicable it is, then you can say, oh, it's not because of random events, not because somebody did it wrong. That's why you got a result. So that is why when it comes to clinical trial, the study protocol, you need to publish it beforehand. And every time you do something different, you have to submit a modification. And then you follow it strictly. So let's go a little bit deeper. In areas in the intervention group, a study team applied this disinfectant and allowed the area to air dry. And they, according to manufacturer's instruction, so that they even mentioned that they did it properly, not just the way they wanted. The areas in the control group received no intervention beyond routine disinfectant. The environmental services team was blinded, right? Again, this is important. We don't want the cleaners to be doing something different and cleaning more carefully just because they think it's a study. All study occupants were occupied by patients with contact precaution. And then they did the regular terminal disinfection using bleach. Here, I'm not going to go through deeper, but basically what they did was clearly laid out all the steps required. And for me, I'm not an environmental scientist, so I don't know the full steps. But imagine if a person who is in this department and want to replicate this method, then you want to, oh, okay, first they did this, then they did that, then day 2, they did this. Okay, so you want to lay out as clearly as possible. Now, again, this another section. I'm not going to go through deeper because I'm not a microbiologist, but here again, you can see they describe step by step how they collect data. How do they make sure that you get the swab? How do you save it? How much, right? Because again, you want to make sure the standard is the same in this situation, and when other people replicate it, they can use the same method. And it has to be accurate too. Okay. So here, for this study, we define. I'm going to highlight this word define. As you read research paper, you're going to start noticing the 2 main things that are really important: definition, measurement, and outcome. Why is definition so important? Because again, we want to make sure everybody knows what you're talking about. We don't want you to talk about a Staph aureus in one study, but another study we're talking about a different type of bacteria. So you want to define it as clearly as possible. An outcome, right here. How did they define the outcome? They define the outcome total contamination. And then how did they measure it? They measure by colony forming units on study day 1. So it's very specific. And then they compare the results from the intervention group will compare against the results from the control group. So here we talked about it in the abstract. I'm going to skip right through. And here, statistical analysis, because it's a randomized controlled trial, there's not too much they talked about. What I wish they described more was the power of the sample size, because not everybody knows what is needed, how much CFUs is needed to get a positive result. So if it would be nice if they included a power calculation here. So now let's go into the results. And for results, I am going to go straight into the tables. Okay. Oh, wait, actually, let's look at the consort flow diagram. In all randomized control trial, you will see these sort of diagrams, and it's really helpful and to see how many groups are in each. There are 50 study rooms, and they randomize each site, right? 50-50, and then 50 analyze. So it's very equal. Now, sometimes when I do journal club with nephrology fellows or residents, when they say, oh, they're only 50, like the sample size is so small. Remember, the sample size is not based on the number of patients. The sample size calculation is based on the number of outcomes and events happening. So the true sample calculation, we need to know what is needed, how many colony forming units. That is how we calculate our sample size, not by the number of study rooms, not by the number of patients, not by the number of sites. Okay, so remember, when you think about sample size, it's not the patient, but the actual event and outcomes. Okay, now let's look at the patient characteristics. Here, we want to see a quick scroll. Seems like a reasonable patient, but I remember somewhere we talked about the patient being bedridden here. 78% bedridden, 68% needed a catheter, 4% needed a rectal tube, and 12 had nothing. So being bedridden, I think, may affect the study a little bit because somebody may not move as much, and so may not have that much. It does not. The person may not touch the surface as frequently. Okay, now in terms of results, so remember we are talking about CFU, right? Let's focus on a p-value here. Of all the outcomes, they found that in day 0, there's no difference. Study day 0, there's no difference in the CFU units between the two groups, which makes sense, right? If you don't do anything, it should not have any difference. So that is important. If you see a big difference, that means your baseline cleaning may not be accurate. Then it's study day 1. This is after 24 hours. Here you can see the intervention group, the one that they cleaned the surface more, had lower colony forming units. And you can see the p-value was less than 0.05, and the 95 confidence interval. Yeah, okay, yeah. So p-value is less than 0.02. I want to bring your attention to this p-value was the whole room. But if you look at each section where they actually swapped that pathogen, it seems like only the sink was the main driver, right? If you look at the p-value here, it's the sink that was contributing to the difference. Bed rails, not much. Over bed table, not much. So if you can think about it, if a patient maybe because they're bedridden, they're sitting there for a long time, and then they are not moving much, and then the sink, I don't know, maybe it's the nursing who comes in and do a lot of changes. So that's why you see more difference. This is just a hypothesis. I don't know, but that's how we think about the discussion section. Like, what could possibly contribute to this difference? I'm not going to go into the secondary analysis as much. Okay, so let me go back to this. And remember, we were talking about the methods. The population seemed reasonable. The outcome was accurate. Now the question is it clinically meaningful? So here they mainly talk about like the first step, whether the bacteria is different or not. But to say, oh, this is the best study in the world, we don't know yet. Okay, because it doesn't really, they're not looking at patient outcomes. So that's one thing you need to put at the back of your mind. Since this is not a large data set analysis, there was not as much on variables and statistical analysis and sensitivity analysis. Any ethical concerns? They earlier in the paper, they talked about getting IRB. They did not get patient consent. They got a waiver. Now, let's go to results. Were the results clearly presented? Was it accurate and in a clear order? So in the table, I like the way they structured the table. It was so clear, you can see study day 0, intervention group versus control group, and then they have the p-value, and you can look at the primary outcome, and then they put under all the different little subsections and the secondary outcomes. So that, I would say, they pass very, very well done. Have all the relevant data been included? Yes. Is the data described in the text consistent with the data in the figures and tables? This is more a reminder for myself as a reviewer. When I look at a paper submission, I want to make sure it's consistent. But what for all of you who are doing just regular critical appraisal, you don't need to go to that detail. The tables, is it easy to understand? Does the table headings make sense? Yes, so far so good. Do they include measures of uncertainty? Right? They have p-values and numbers. You can't just say, oh, group 1 is better than group 2, and you don't include the p-values, you don't include anything. So it's important to include all these information to show how much uncertainty there is. The figures, was it easy to understand? Yes. Do the figure legends make sense? They did not have that many figures other than a consort table. Okay, so this is the first level of how I look at research papers. I'm going to go briefly into the discussion section because it's pretty long, and I will go straight into the limitation because this will see how the researchers think, the authors think about their research, and then see if they have thought through the bias and see how they fix it. Okay, this study has limitations. First, the disinfection of clinically relevant spore-forming bacteria was not assessed, given that these quaternary ammonium compounds are not efficacious for these pathogens. So they are clear to say that, okay, it only disinfect a certain type of bacteria, but not the spore-forming type. Second, only rooms occupied by patients with contact precautions were enrolled. So yeah, I was wondering about that. Why only contact precautions? These special patients that have big, bad, terrible bugs were enrolled. This eligibility requirement may have set a higher bioburden baseline on day 0 compared with non-contact rooms. So they are choosing a very subset of patients. These are patients with already a lot more bacteria. So as they say, they have set a higher bioburden baseline at 0 compared to non-contact room. When you have more bacteria at baseline, it's more likely to see a difference. So it may only, all these special disinfection wipes may only be effective if there's a lot of bacteria and you can see the difference. But in regular floors, surgical floors where you don't have that much bacteria, may not even make a difference, right? So they are bringing this up. Moreover, 78% of patients were bedridden, which may have affected the environmental contamination of sample areas, such as inflating bed rails and overbed table contamination, as well as deflating sink contamination. I don't know what this deflating sink means, but I'm assuming they have a special sink or something. But I think what I mentioned before, because they are bedridden, it means that the contact surface may not have the touching of surface may not have happened as expected for other patients. Third, enhanced disinfectant was applied by the study team, and most patients were in the intensive care units. As a result, generalizability to other settings may be limited. And 4, patient acquisition and clinical outcomes were not measured. Okay, so these two are really important. That's generalizability. When we think about randomized controlled trial, the first step is understand how effective it is. Okay, A versus B. Oh, okay, yes, we see one is better than the other. Then we want to see whether this result can be applied to all different settings. We think about, okay, what do you mean by setting? Okay, so this is done in ICU in a specific group with contact precaution. So that means maybe other ICU, regular patients, ICU, but not in who don't require contact precaution may not be effective. And then next is floor patients, it may not be effective, right? So that means the results only apply to that small subset of patients. And in the future, if we want to do trials, we may have to do a trial on different settings to test it out. Fourth, this is the most important thing. Patient acquisition and clinical outcomes were not measured. I'm not saying that every study design have to go all out, but usually this is the first step, just seeing whether you can even demonstrate some bacteria. Randomized controlled trial that have more funding, if they can, they try to do both at the same time. They actually do the colony forming unit, and then they have extra resources, and then they will track the patients, see what the outcomes were, and then they follow them up. So that's why certain the number, all these information, adding bio information, add clinical outcomes, adding long-term outcomes. If you see these sort of studies, these type of clinical trials will get into high impact journals. So that is a difference when you think about why certain why certain randomized controlled trials get into huge, like NEJM versus this is a good one in JAMA too, but there are some who only get into open access journal. Is because of the quality of the study, how you are linking your question to the final pathway of patient outcome. Okay, so it's 30 minutes. I've put so much information to all of you. If you have any questions, you can put it in right now. I'll wait for 1 or 2 minutes. This is the start. I will continue doing more live streams on different type of study designs. This is a fairly simple design, and the analysis is clear cut. Sometimes you have to think about randomized controlled trial where they have 3 different arms and then they switch over, and sometimes they also have different analysis per protocol versus intention to treat. So in the future, I will dive deeper into that type. And the key point is when you start reading a research paper, any term you don't understand, look it up and find the actual definition and understanding it. This is how you start. And the way I learn different research methods is basically just read this and replicate the step. And the more you read it, you realize, oh, they all use the same steps. And so thank you so much for all of your attention, and I will see you very soon again. Take care. Bye.